Thank you. So our goal today is to continue talking about the program, the Metagenomics, OmicsLogic Metagenomics program. And we'll talk about it starting from kind of the beginning. So last time we did an overview of the program. We talked about what types of applications of metagenomics we will be looking at. And we talked a little bit about the structure of the whole program. Today, what we'll do is we'll start talking about how we can study microbial communities using sequencing. So we'll give a few more minutes for people that are joining to join the program. And in the meantime, what I wanted to go over are some of the technical details of the program and how everybody can register on the platform and get access to the courses and to the platform that we'll be using throughout the course to analyze the data and learn about some of these examples. So let me just show you a few links that you probably have already seen. We have a website called EDU t-bioinfo, which is an educational website where you can gain access to a lot of free materials, free courses to get training, and to learn about some of the background information and the terminology that we'll be referring to. So I encourage everybody to join and create an account to show you basically once you create an account, you can go to your profile, and under the profile, you'll be able to see your progress. So the main point of this profile is to assemble the information about you, and that information will follow along with the different activities that you perform. For example, if you finish or register for courses, if you complete quizzes, if you do the practical assignments, all of that information will be available right there under your profile, and you'll be able to see where you are, when you enrolled in different courses, and where you are today. The goal of this profile is to eventually help you understand your journey and share your journey with others that are doing similar things. So if you're looking to advance in your career, if you're looking to get certifications, you get certificates for these courses that certify your participation and mention what topics were covered. You will also be able to see how you perform compared to other people. And an important aspect of this is that the participants of this program, those that have registered, are going to all be listed under the organization that we have for this topic. So let me quickly show you. So this is the page for the organization, and the page kind of assembles all of the information that you will need to benefit from the program. So what are some of those things? You will see that we have the kind of upcoming events. So once you register and become a part of this program, or if you want to register for events individually, you can scroll through. You can see our schedule for upcoming events and what each event will be covering and the date. You will also see some of the activity of participants here underneath. You will also see here are a couple of courses that you would benefit from taking if you are new to metagenomics. We have right now Introduction to Metagenomics and Metagenomics 1. And in the process of this program, we'll be also adding Metagenomics 2. So the goal of these courses is to supplement some of the materials that we'll be covering in these online meetings, online sessions. And you can really benefit from going through those and seeing some of the examples that we'll be discussing. You can also see here under curriculum how these different courses and events fit together for the full curriculum of the program. So all you need to do is go there. Now, what we are trying to do is get together a group of people that are together studying a variety of different topics in metagenomics and can learn by practically applying the learned skills to data sets. And these data sets are going to come from public domain, from publications. So we will show you, walk you through how to also identify new data sets that you can analyze using various methods that we will be discussing. In parallel to this educational portal, you will also get access to the t-bioinfo platform. The t-bioinfo platform was designed by the Tauber Bioinformatics Research Center, and a version of this platform is designed specifically for education to go along with some of the course material that you will find here. So what you will find on this platform are publicly available algorithms that are packaged inside a visually intuitive interface. So this interface really is a pipeline builder and a version of pipeline execution script that essentially allows you to run one script, take the results from that script, and pass it on to the next script after that. And we'll learn a little bit about some of these sections. In particular, we'll be looking at a couple of examples here under metagenomics microbiome and amplicon 16s and 18s sequencing analysis. So what we'll be doing is we will be covering some of these sections, but by participating on the platform, on the educational site, what you can do is you can go right here under access and fill out your details, specifying here what is your intended use. So for example, if you're interested to use the platform for education, you can say, I am a participant on the educational website, and I would like to apply the learned skills to a data set. So I would like to use it for education. If you're interested in using this platform for research, you can also request such access, which is a different type of a license that you get. If you're not sure and you're just exploring, you can also request a demo account just to see how it works. And once you get a demo account, what you'll be able to do is you'll be able to use some of these examples that are preloaded on the platform, and play around and understand how this pipeline works. So that's a little bit about the program. I also wanted to make sure that everybody understands that the program itself is going to last for a couple of months, November and December. And during those months, what we intend to cover are a variety of topics related to metagenomics. So we will discuss very early kind of concepts like what is the microbiome and why do we use sequencing to study the microbiome? What is next generation sequencing and how does it apply to metagenomics data? And we'll talk about that today. But we'll also move on forward from there and start talking about some of the analysis methods. So how do we analyze different types of next generation sequencing data? That type of analysis could be applied to different types of applications. Detection of some microbes in samples by separating the host and the pathogen, and then looking in a variety of databases to spot which pathogens are present. We'll look at metagenomics in the context of trying to, a priori, look for whatever is in the sample without knowing what exactly we are looking for. So not looking for some specific pathogen or some specific bacteria, but looking what is in the sample and trying to analyze the sample from that standpoint. And also we'll look at how we can study the taxonomy, the proportions of different types of organisms, composition of the microbiome, and be able to compare different samples. How do we understand the composition changes from condition to condition? We'll also try to understand, interpret what might that type of analysis reveal to us. And we'll look at it from several standpoints. We'll look at it from the standpoint of microbial drug resistance. We'll look at it from the standpoint of viral evolution, and how viruses evolve to overcome a series of challenges that they encounter due to the immune response. And we will also take a look at other types of projects like the rhizobacteria in soil to really see the diverse application of metagenomics that are applicable to this type of analysis. And we hope that all of you will benefit from joining for this kind of a journey, exploring together with us, and having a discussion. So one of our main objectives is to really facilitate a discussion about how might you be interested to apply such techniques to a topic that is interesting to you. So we encourage all of you to start thinking about some of these ideas. If you struggle with any of the terminology and the topics that we're covering, we are encouraging everybody to take some of these free online courses and review the material that we have there. And the sessions will build on that material, hopefully allowing everybody to get direct, hands-on, practical experience on analyzing some of these types of metagenomic data. So how did we get there? How did we get here? And by the way, I'm going to be leading this session, but I will be, the whole program will involve other speakers as well. And we will also allow for some Q&A sessions where you can ask questions. So if you have any questions, please make sure that you put those questions in the chat box, and I'll be happy to address those. So in today's lecture, we want to talk about how did we get here? What is some background information? Why are we even turning to sequencing to study metagenomics? And then we'll talk about some of the sequencing techniques that allow us to, and what do they allow us to study essentially? What kind of data can we generate? So when we talk about how do we get here, we're really talking about the history of metagenomics. Now, metagenomics sometimes is confused with 16s sequencing, but in reality, metagenomics is a field of study, the field of study of the microbial community using genomic information. So when we speak about metagenomics, we're really talking about historically, when the microscope was invented, it was invented to spot some of the smaller things, the microorganisms and the cells in bigger organisms. And as that technology started to evolve, we started discovering that there's a whole world of microbes. There's a whole world of tiny little things that live and work together as communities. And so one of the first things that happened right after that is that we discovered that these microbial communities could be grown. And so here you see a culture of bacteria in a petri dish, and you also see different examples of microorganisms that could be seen in a microscope. So what we can do, one of the things that could be done, right, is we could extract a specific strain of a bacteria, grow it in a lab, and then we can look at how does that bacteria grow in different environments, and how does it interact with a variety of different influences, right, if we change its environment. So those types of studies are very limited because, of course, to do that, we have to extract the bacteria from its natural environment, from where it is naturally, place it in an artificial environment, and then perform all kinds of tests. Not only that, it is also believed that only 1 to 2% of all bacteria could actually be cultured in a lab. So if we want to study how the bacteria grows, how it works, how it communicates with other bacteria in the microbiome, we really have to have an alternative method of study. And so as a result of that, this explosion of sequencing or high throughput sequencing technologies, and by explosion, I mean the amount of sequencing data that is being produced due to the lowering costs, due to the accessibility of this technology. Now we have this whole field of study called metagenomics. And so for any one of you that is interested in metagenomics and kind of how did we get here, what are some of the opportunities and challenges in metagenomics, we created a short crash course called Introduction to Metagenomics. So you can go to the website, and once you are registered, you can go to a collection dedicated to metagenomics, where we have Introduction to Metagenomics and Metagenomics 1. And two of these courses are a very good start for anyone who is just new to this whole area and wants to understand a little bit of where we are in the field of metagenomics, and learn also about some of the ways that data is being collected and analyzed in metagenomic studies. So in Metagenomics 1, for example, there are already some examples and in detail a review of different sequencing techniques. How does sequencing work? What are some example projects? Where to find this kind of data, etc. So for anyone that is really starting, I highly recommend to take some of these quizzes and test how you understand the materials based on some of the quizzes that are offered throughout the course. So I really encourage all of you to go through that material, and that will also clarify any kind of questions that you might have in terms of the terminology that we use, because a lot of times what will happen, and I'll just give here a quick example, is you will actually be able to hover over some of the terms that are mentioned that maybe are not very clear. Maybe we don't spend enough time to explain those terms. But you will be able to hover over those and really look at what they mean, get a definition. So I really encourage all of you to go through that, read through, hover over some of these highlighted words, and understand what they mean. So what we'll be doing today is we'll be actually going through some of the material that is already covered in these courses as well, but talking a little bit more in detail about 16s metagenomic sequencing in the context of next generation sequencing in general and how it applies to the microbial study, to the metagenomic studies. So if anyone has any questions, again, this would be a good time to ask those questions during the session. And by questions, I mean anything that is not clear from the material that we're going through, anything that is not clear to you about the program itself, or anything that's not clear to you from the types of projects that we are discussing. So really, if you have any of those questions or concerns, please feel free to add them in chat and we'll be happy to answer them. So this session is really going to be dedicated to 16s metagenomic sequencing. But before that, I want to start with a little bit more background about sequencing itself. So what's unique about metagenomics is that as all the other omics, they are really focused on a single organism, right? So if you think of genomics, proteomics, transcriptomics, they all focus on the whole of a single organism. Or today you also have single cell sequencing technologies. You can look at different types of cells through cell sorting, or you can look at different tissues, or you can look at different even individual cells, right? But still, we are going to be looking at a single organism. But organisms, especially microorganisms, live in closely related and tight-knit communities and interact, and in fact, their function oftentimes complements, and they complements each other and also the host that they live in. And so when we want to study such a community, we need to look and study and understand the genomes of individual representatives of that community. So metagenomics is the study of the metagenomes, the genomes of those organisms. So we look at the composition of that community by looking at the genetic material that is directly taken from the environment. So this is something that allows us to bypass this need to take a single representative and culture that bacteria in a lab. And it allows us to maintain kind of the natural environment, but also the composition, because it's important, the proportions of different types of bacteria together, and also different types of interaction. So we can really look at that community from a number of different ways. So that's not to say that we don't apply sequencing to cultured bacteria. So if you look at, and here we'll start talking about some of the challenges as well as we kind of look at this. Here you see the gut, and you want to, for example, study the bacteria and the microorganisms that live in the mucus around the gut cells. So to do that, obviously, there are going to be several challenges. How do we separate what is in the mucus from all the other cells? You have the immune cells, you have the gut cells, you have what's inside the gut. So there's a lot of things that you are actually sampling. And there are two ways that you can do it. You can extract the mucus and you can culture an individual strain of bacteria and grow it in a petri dish in a lab, and then sequence and understand what's going on. Or you can take just the full sample and then culture-free sequence that material, selecting, now you want to select for something very specific. So we'll talk about how do we do that. Before I move on, Nadeep is asking questions, so thank you, Subhrajit, for answering that question. So what we see here is really what are some of the results that we can obtain, right? And these are the two types of things that we might be interested when we are looking at metagenomic studies. We can look at classification, right? So composition of different representatives inside this community, inside the sample that we have. Or we might be interested in looking at complete genomes. What types of genomes, so the full sequence of that genome is important, but to do taxonomic classification, the full sequence of each individual is actually not that important, right? And we'll talk about how to actually do that. Now, in terms of sequencing, the way to prepare that data, right? When we take that data out, we then have to produce digital information about the sample that we have. And that digital information, since we're looking at the genetic material, we need the sequences of DNA or RNA that is present in the sample that we have. So here are some examples of machines that produce this kind of sequencing information. MinION is a handheld device where you can put a sample and it will produce the sequence. You have Illumina that allows you to fill a whole number of slots with material and sequence it. And then also PacBio, another example. And so all three of these examples are used for metagenomic studies. There are differences, however. You have long reads and short reads. And we'll talk a little bit more about what that means. But essentially, we're talking about some benefits and some downsides to both types of technologies. Both MinION and PacBio produce long reads. However, the quality of those long reads is going to be lower. So the reliability of each individual nucleotide is going to be lower because we gain in length. However, if we are looking to assemble full genomes, the longer the read, of course, that's going to be easier to assemble. We have less pieces to assemble together. They are longer, so we can more easily combine them together. Illumina, however, provides higher coverage, and therefore we have higher confidence in the sequence of individual reads. And it's a much more established technology from the standpoint of reliability and availability. And it's not as costly. So in fact, from the cost perspective, MinION would be the cheapest. Illumina is fairly affordable and very widely used because of that affordability. And PacBio tends to be a higher quality, so more reliability than MinION. However, it is typically more expensive. Now, another benefit of some of these newer technologies like the MinION is the time that it takes to produce a result. And so here's an example of a publication that compares how a protocol using MinION, Oxford Nanopore MinION, compares to the MiSeq Illumina sequencing from the standpoint of time and from the standpoint of steps that you need to take to get to a final result. And now when you think about this, and Debanita will have a chance to talk about PacBio a little bit more, but right now, what we want to get to is the 16S ribosomal RNA sequencing. So that's what this session will be about, but I'm happy to share more materials about PacBio as well. So when we compare long read sequencing on something like MinION, which is a handheld device, so you can really take it anywhere, and also it is cheap. One of the major benefits is the time that it takes to identify what is in my sample. So if we think about rapid detection of viruses or pathogens or characterization of what causes a specific effect, this is a very useful method. You can see here that essentially in a couple of steps and in less than 6 hours, you can get a detection, right? Typically what we mean by detection is presence or absence of specific types of pathogens. In Illumina, you're talking about several different steps and over 20 hours that are needed to get to a final result, right? So that could play a big role in your selection of technologies. But to understand how these technologies work, and knowing this example of sampling from the natural environment, let us talk a little bit more about how 16S ribosomal RNA sequencing works. And thank you, Subhrajit, for putting that link in there. So anyone that's interested specifically about PacBio, they have some details available on their website. And if that is a topic that's interesting to more than a few of you, we're happy to schedule a session, maybe discuss some of that technology as well. So 16S ribosomal RNA sequencing. 16S ribosomal sequencing is also called amplicon sequencing. It is amplicon sequencing because it amplifies a region of ribosomal RNA. And so when we talk about Illumina sequencing, we're really talking about several different options that are available. And there we are looking at the differences from why we are doing the sequencing. In other words, what is the objective of our study in terms of cost, how affordable, so maybe that means how many samples we can sequence, and also talks about how detailed do we need to get our result. So how much detail are we looking for? 16S ribosomal RNA is one of the lower cost, and it is also typically used to understand taxonomic classification and composition of a microbiome. Full-length sequencing, 16S ribosomal RNA full-length sequencing, meaning we are sequencing the full 16S ribosomal RNA. When we talk about amplicon sequencing, we're talking about amplified regions of that sequence. And 16S ribosomal RNA shotgun sequencing typically requires higher sequencing depth. And the goal from the full metagenomic shotgun sequencing is also something that is possible with Illumina sequencing. And typically those would be used for assembling. So full metagenomic shotgun sequencing would be used to assemble full genomes present in a sample, which is, of course, much more challenging than trying to look for this 16S region. Now, what is the 16S region? 16S region is the ribosomal RNA, and this is characterized, we'll talk in the next slide, this is only specific to prokaryotes, so they're not going to be included in the host, right? If we talk about a sample coming from a human or a mammalian host, we're going to be taking a sample, and this is an easy way to separate the prokaryotes in the sample from any other type of genetic material that we have. So in a way, it is really a useful way to separate and separately look at just the prokaryotic, so only the microbial representatives, individuals in the sample. But not only that, we also have primers that could help us detect just the 16S sample. And so you can see right here, this is that 1500 base segment that could be amplified with PCR. But also we can actually look for some specific regions and only look at that region, and that will help us detect the taxonomy of our sample in general. So we'll talk about that in detail here in a second. What is ribosomal RNA? So the ribosome, of course, the ribosome, of course, is a protein that functions to, yes, a complex that produces proteins. So it goes to the RNA, mRNA, and we know that the prokaryotic ribosome contains two portions, the large subunit and the small subunit. And so what encodes for the small subunit is actually the 16S ribosomal RNA. And so within this RNA, this is what it looks like. We have multiple what is called hypervariable regions. And these hypervariable regions are actually what we can use to detect specific level taxonomy. So if we talk about how we can use this material, we typically select for 16S ribosomal RNA, and then look for these specific V1 to V9 conserved regions, which are between 30 and 100 base pairs long. And then just those regions, right? So sometimes several of those regions are assembled together in a single Illumina run. And so here you can see how you have a sample collection, and then RNA is isolated, or DNA could be isolated, but in our case, we're talking about RNA isolation. And then you have the 16S ribosomal RNA gene that is amplified, for example, for the V4 hypervariable region. And then once we have that raw material, we sequence that material that then produces a specific type of data. Now, another thing, so I see here some discussions about primers, and so all of that falls under the category of library preparation. So all of this discussion up until now is really about sample extraction and library preparation. Just to give you a little more detail about that, some of the kits that are typically used are MoBio DNA extraction kit, QIAGEN DNA microbiome kit, and there is a number of other kits that are specifically designed to extract this type of material from your raw material. And also for library preparation, this is actually going to be important because one of the biggest considerations for library preparation of environmental samples for shotgun metagenomic sequencing, like we talked in the previous slide, it has to do with this amplification. What do we amplify? And so certain types of samples like water or swabs, they only yield small amounts of DNA, and so we have to amplify the material that we have during library preparation. And amplification by PCR can actually over-amplify some of the material that we have, right, without our control, and that typically can yield a distorted view of the taxonomy that we're looking for. And so we often need to kind of make these decisions when we have low inputs of DNA and to minimize variability. And so, you know, we typically want to use amplification-free library preparation methods if we have low yield of material in our sample. And there are several kits also available for this, including the Bioscientific NextFlex PCR-free DNA sequencing kit, Illumina TrueSeq PCR-free library preparation kit, and CAPA Hyper Prep kit. So there's a number of kits that are specifically designed for this. Now, whenever the sequencing itself happens, whenever we talk about Illumina shotgun sequencing of 16S ribosomal RNA, we would typically use paired end reads. And paired end reads, as you can see right here, this is on the flow cell, they are really sequences of the same element of RNA, shattered RNA, that are read from both sides, from the 5' and the 3' ends. And when they are combined, that gives us approximately 250 to 300 base pair long segments. And so these are longer longer reads, and we have multiple representatives of the same region because we have high coverage. And because of that high coverage, we need to merge them together by looking at just the ones that actually have pairs. So they have both the forward strand and the reverse strand reads. So we need to eliminate those that do not have those pairs and then develop a consensus. So where do the sequences agree in order to organize them into these organizational taxonomic units. So to understand what that means, let's talk a little bit more about what is taxonomy. So taxonomy refers to the classes of different organisms that are present in our samples. So for example, when we talk about microorganisms, we're talking about a diverse population, so to say, but the lower the level, the more diversity we will discover, right? So one of the useful things for us to think about when we do this kind of sequencing, when we do this kind of analysis, we really want to look at how much diversity do we need to be able to answer a specific question. Okay, so a question really quickly, what is paired end reads? As you see right here, individual reads are segments, short segments of sequences. So when we say shotgun sequencing refers to shattering the RNA that we have. Typically, then those pieces are going to be sequenced. And so we have this digital information about each read. And so we can just take them all from one direction, or we can take each one of those short segments and sequence them from both directions. And when we sequence them from both directions and align them together, we have a higher, more reliable sequence that we can use in our downstream analysis. So that is referred to paired end reads, and we typically use paired end read sequencing when we use these kinds of 16S metagenomic studies. So a question here on how to optimize the amplification of the 16S gene prior to next generation sequencing. The bacterial 16S gene contains 9 hypervariable regions. They all range between 30 and 100 base pairs long. And they're all involved in the secondary structure of the small ribosomal subunit. And they all have a degree of conservation that varies between these hypervariable regions. And so the entire 16S sequence allows us to compare all the hypervariable regions. And so if we take the whole sequence, it's about 1,500 base pairs long. But sometimes that's too long for studies that want to just look at bacterial community composition. And so when we talk about the Illumina platform, the reads are much more cheap to produce, but they're not that long. And so that's why we have up to 300 base pairs typically in a read. And so that's why you want to sequence the full, the hypervariable region, right? So those regions, again, range from 30 to 100 base pairs. So you kind of have these different options, right? May, I think I'm kind of taking a long route at answering directly your question, right? So one is we can just amplify the 16S full gene, right? So if we go back right here, we can actually use PCR amplification to just focus on 16S gene. Or we can actually look at the specific hypervariable regions, right? And again, we will use PCR amplification with specific libraries for those hypervariable regions. So we can look at all of the hypervariable regions, which is all 9 of them, or we can look at one of them or a couple of them. And so they all have different degrees of conservation. And so more conserved regions correlate to higher level taxonomy and less conserved regions to lower levels, such as genus and species. So again, it's important to look at this. How do we want to study? How much detail do we want in our resulting data to be able to answer some of these questions? Okay, some more questions. In the slide, I can see at the third step, it is written PCR amplification of V4 region. What is the significance of V4 over others? So the V4 region is actually one of the more conserved ones. So we can use that region to place our characterization of the sample composition within a certain level of taxonomy. V3 for region, I have the Illumina protocol, but it does not work for all types of samples, especially with low amount biological fluid. Especially, so as I mentioned, there are some specific types of samples that we're talking about, if they're from water or from swabs. So you don't have a lot of RNA, sufficient RNA or DNA to capture, what's happening, right? So if you have again, we can go back to the slide right here, right? When we have a swab, we're really talking about taking the mucus, and then we have a limited number of that material to be able to extract what we're looking for. So we can actually, think of the PCR route when we amplify the material, but that amplification, unfortunately, is not going to uniformly amplify all of the present data, all of the present RNA. So if we use a non-PCR amplification library prep, it's not going to have sufficient material sometimes to study everything that we might want to study. So that's briefly to answer your question. Any other questions about any of this? Is duplication or triplication may create error during process and specifically during data analysis? Exactly, yeah. So that is one of the things that we'll start talking about next time. We'll talk about how do we apply this technique right? So now you understand that when we talk about metagenomic sequencing, we're actually talking about many different types of sequencing. We're talking about many different types of instruments, so at least several types of instruments. When we talk about different strategies to produce our data, we need to make a decision on which instrument is going to be used. We're going to be looking at which read length, asking 252 base pair is the length of a read. And we're going to be looking at different strategies of collect data, instrument that we choose, library preparation, read length, and then that will define what kind of analysis we are going to be trying to perform. And it will also be defining how can we utilize that analysis result? What kind of information can we get out of that result? So as an example, let's take a look here. What we are looking at is biological classification, which is taxonomy of these microorganisms. What kind of taxonomy can we use to characterize a community, right? Now, what is a community? A community is going to be a microbiome, so what we expect to find in one of these areas on the human body, for example. Or this could be in the environment, from the soil, from the water, etc. So when we look at those, we are looking to study diversity. Sometimes we just want to look at diversity, and that would be an example of using 16s amplicon ribosomal RNA, because we're going to be looking at amplified V4, for example, regions, or V4 and V3 regions. That will help us define, just compare this type of what's called alpha and beta diversity. So how much, based on species, for example, we have, and then we can compare them and understand the differences. However, sometimes you want to look at, for example, presence of a specific detection of a specific organism. For example, a virus. I want to know whether there is a specific virus and how much of it, and what is the diversity of that specific virus. So if that is my question, then I cannot use 16s sequencing for that, because I am going to be more interested in the full genomes of that virus, and I want to study in detail the differences between those genomes, between the sequences of those genomes. And a virus is, of course, going to be maybe several thousand base pairs long, maybe 15 or 30 or 40,000, but we will also look at the sequence itself and not how diverse a sample is. So very different strategies are designed around the analysis needs and are designed around the application, the use of that analysis. Okay, so another question, how to optimize the amplification protocol regarding the number of amplification cycles and input DNA. So what may be a good discussion for us to have is to better understand exactly what is it that you are trying to sequence and what is your objective. So what I propose is for you to maybe, if you are interested in analyzing this kind of data, first of all, you can write your question directly, or you can also go, as I showed right here, where you request access, and you can write your type of a project that you're interested in, and we will be happy to address any of your questions, maybe more in a one-on-one type of a discussion, if that makes sense. So again, the shotgun sequencing is unique in the sense that we're trying to sequence a large and diverse pool of microbes. Each one of them has different size, and a lot of times they're mixed with host DNA, right? So when we talk about 16s sequencing, we are really talking about being able to separate 16s ribosomal RNA, which is only going to be for prokaryotes. And we then look for a specific region. So we look typically in the hypervariable regions. And then we have high coverage. If we use Illumina, we have high coverage for those specific hypervariable regions. And we are able to look at the taxonomy of our sample. I mean, how to optimize the amplification protocol regarding the number of amplification cycles input. Yeah, sorry. Yeah, so I hope that makes sense. Now, when we look at the analysis after that, right, what we wanted to talk about today was that, okay, we understand what we are sequencing. We're taking the encoding region for this small subunit of the ribosome, and we are taking these specific hypervariable regions that are going to be involved in the secondary structure of the small ribosomal subunit. So because the ribosome has to be stable, these regions are conserved. And they are conserved so that we can use that conservation so that we can study taxonomy. Conversion into reads cannot differ in duplicates or triplicates, and if it differs, then there might be error. Is it correct? So you're talking about pair and reads, as I understand, right? So pair and reads, yeah, if you have, two examples of the same thing, and you can see a change in just one of the copies that you have, right? So you have two copies. If you have a change in just one copy, then you know it might be a mistake. But if you have a change in two copies, that gives you a higher confidence that you are finding a real change, right? I'm talking about in the context of now aligning those reads to the reference genome, right? So whenever we will talk more about, so after this, and maybe this is a good transition to this final slide, we will talk about better understanding the microbiome. So we'll actually look at some examples where in the context of those examples, for example, when we look at gut health and the brain or the gut microbiota, right? When we look at this example of what we saw here, right? These different types of microbiomes, we'll talk about how these different considerations about what are we sequencing, how are we sequencing, and then ultimately that will dictate how we're going to be able to analyze the data that we have. So we will talk about, in the context of very specific projects, thinking about the objectives that we have in each project, we'll be able to see how we can leverage this type of a technique of 16s hypervariable, meaning amplicon sequencing, how we can leverage it in these specific types of projects. So again, what I wanted to encourage everyone to do is, and I'll talk a little bit more in detail what we're going to be doing next, but I want to kind of summarize what we covered today. We covered today how we can, in the context of this program, any one of you can get access to free materials if you have some of these questions. In other words, how is the sequencing done? How do we take samples, prepare those samples for sequencing? What is the process of sequencing? And what we recommend are these two courses that we prepared specifically for that: Introduction to Metagenomics and Metagenomics 1. So if we just take a look at Introduction to Metagenomics, some of the material that we covered today, we'll talk about some of these strategies. So you would be able to learn about the different types of metagenomic sequencing, 16s amplicon. Kind of if you don't understand some of the terminology, again, you can hover over and learn about different types of sequencing. And then also, if you take the Metagenomics 1 course, the Metagenomics 1 course will also cover some of that information. More for logical steps of different types of metagenomic sequencing and things like that, including some example data sets. So you can actually look at some examples of where 16S ribosomal RNA is used and some places where the full assembled metagenomic sequence is important to analyze the data. Okay, and also some of these materials will talk about the biological background and methods, something that we also covered briefly today. Okay, so here you can learn about the 16S ribosomal RNA, some of the extraction techniques for either 16S or whole metagenomic sequencing. You can read about the reads and what is that, and you can also see some of these terms that might not be very clear, and really learn about different types of metagenomic studies. For example, what we won't talk about in detail in this program are metatranscriptomics, biochemistry, and other types of important aspects of microbial genomics and microbial studies. Okay, so again, what we will talk about in our next session, we'll be talking about how 16S amplicon sequencing is useful to study the microbial communities and their impact specifically on human health. So we'll talk about examples with the gut-brain axis, where we'll look at changes in composition between different types of diets and also the impact on behavior. And we'll also start talking about an example where we can look at how diverse gut microbiome this will give us insight into how we can leverage some of these techniques to analyze different type of data and study the microbiome, but also just to use microbiome as a means that will be important in the future aspects of our discussion. So next session will be focused on the microbiome. We will then talk about the analysis, some of the challenges with the analysis, the challenges of short reads, and what we discussed today with the paired end reads and the hypervariable regions and all those things. What are organizational and taxonomic units, and how do we actually make sure that they are accurately represented in the analysis? We will have then another session to map the data that we onto databases. And what's going to be important for us is to try to understand the presence of a variety of different microbes or microbial species by looking at databases with annotated hypervariable region sequences. So actual full sequences of these hypervariable regions in different databases like Silva, Green Genes, NCBI, etc. And then after we talk about mapping on databases and seeing some of those examples, we'll kind of review the full pipeline. How does a full 16S metagenomic sequence pipeline look? What do we input? What are the different steps? What is the significance of different steps? And what is going to be the output? And what can we do with that output, right? The output could be modified based on our needs. Then after that, we'll review some other examples where we can use full metagenomic sequences of a specific type of a bacteria. So for example, we'll look at microbial response to soil composition, because when we look at just one example, right, rhizobacteria would be an example of when you take a look at just a single type of a bacteria, rhizobacteria, that's it. And you look at that rhizobacteria in terms of the changes in its genomes. So you have these mutations, you have these changes in the genome, and the question is, how do we associate those changes with environmental changes? For example, presence or absence of specific chemicals in the soil. We'll then talk about other examples of metagenomics, so a study of another type of a problem statement where we're trying to detect viral genome presence, right? So if you take a sample from the human blood, for example, or if you take a sample from a culture where you're trying to produce biologics, for example, vaccine production, how do you analyze that data to only focus on non-host, non-human RNA or DNA, and then find maybe traces of some specific viral or bacterial genomes, and then to try to understand what does that mean? Does it mean that we have the full genome? Does it mean that we just have some small segments that resemble some genome, but they're not really a genome? So we'll talk about some of that. So we're really going to be talking about some examples of uses of metagenomics and projects where they could be used. We'll talk about phylogeny. How can we understand the phylogeny or the relationship, evolutionary relationship, divergence based on, for example, specific genes or specific regions? And we'll talk about phylogenetic analysis in the context of metagenomics. We'll talk about drug resistance. Drug resistance is a big problem in tuberculosis, where actually prevention now is one of the main focuses because we have this problem with drug resistance, drug-resistant strains of tuberculosis. And so we'll look at an example study with new data that is being collected recently, and we'll look at how to analyze that data and what types of analysis methods can help us spot and characterize this drug resistance, so understand it also on the biological level. So these are some exciting projects that we'll be covering in the whole program. Again, for anyone that wants to register and participate in the full program, if you're not sure, you can go ahead and start with the free courses that are available for anybody. All you need to do is register for free and take some of the online courses. If you want to participate in events like the one that we had today, work with others on your own projects, develop your own project in metagenomics and see it from beginning to conclusion. And if you want to have a certificate for the program itself, covering all of these different variety of topics, we welcome all of you to go to this link, finalize your registration, and as a result, you'll be getting access to the program, to the sessions that we have, and also we will guide you to utilize the platform and analyze data. So I'm really excited about this program. We'll start meeting again next week. We have a number of sessions coming up that are going to be very exciting. I will, after this session is over, we will be emailing you the recording of this session so you can review some of the materials that we covered. And we'll also share with you the presentation that we have today. And then starting from next week, we'll really focus on those of you that have completed the registration, sending you account information for all of you, and we'll start sharing more data sets that we can start practicing with together. So any final questions, you're welcome to put them in chat. I'll be happy to answer them. You are also welcome to directly contact us through the form that we have on the site, or we have several of our members here in the session. You can email us directly. Okay, so if there are no more questions, then it was really good to see all of you here today. And we are going to meet again next week for those of you that would like to continue joining these sessions. If you have any questions, thank you, Bibsha, for putting your email address there. You're welcome to email us. a question, do I have to pay to attend all the sessions as I have already registered? If you're already registered, then you don't have to pay. Bibsha, if you could maybe check Agni and the registration there. We'll be happy to, if you have gotten the membership for the program, you do not have to register for individual events unless you want to. I mean, you can just click right here on register all events. And if you already have the membership, then you'll be just automatically included in all of the events. And we'll be sending you the login information for each one of our events. If you want to just join one of the events and you don't want the full membership, you can find the event that you're interested in and then register now right here. So if I click right here, each event has a specific, for those that are registered, it's free. For those that are not registered, you will have to pay for that individual event. And then we can send you out the information. We actually have a list of those that have completed the registration. You're welcome to connect with Bibsha to ask any of those types of questions about registration. How do I put my target and current project and inquiries on the website? Well, first of all, you can email us directly. But if you would like to submit a project, an easy way to do that, like I said, is you have this access link right here. So if you go to this link, this is the link where we have our analytical platform. Once you get to that link, you'll see something like this. And right here on the top menu, you have access. So this is designed for those of you that would like to analyze data, even if it's your own data, if it's data that you're planning to generate, and you want some assistance with that data. What you would have to do is you would have to just fill out this form. What is your name, your state, region, city, a company or organization, and then right here, topic of interest or intended use, you actually type in, I am working on a metagenomics project and would like to analyze data. I would like to speak with someone, right, about the analysis and data preparation. Something like that, right? And then include your phone number so we know how to reach you. And then you submit, and we get that email. We review these daily, and then we can get back to you and schedule a time to speak one-on-one and discuss in detail how you are planning to generate your data, what are some of the analysis techniques that might be applicable. If you need help with analysis, we can also help you, but also then talk about interpretation. I think learning about the context of metagenomics in a program like this is obviously a great place to start. Okay, well, if there are no other questions, again, I think we covered a lot today. So thank you again for joining, and we'll be seeing you again.